My name is Shan Brunel. I'm a PhD student in the WARS group at the MRC Center for Medical Mycology. As I just finished my 3-year IMS PhD studentship, supported by the Chloe Fund, I would like to take this time to summarize my work and to give an overview of my project involving Aspergillus infections in people afflicted by cystic fibrosis. Aspergillus is abundantly present in our environment. As a consequence, we inhale hundreds of Aspergillus spores every day. However, in a healthy lung, these are cleared efficiently without any illness. But in cystic fibrosis, they are not, and it's not clear as to why. So, in my project, I have assessed the response of immune cells from CF patients against Aspergillus. Because immune cells from CF patients are missing an important protein, the CFTR protein, they might be impaired in their response against Aspergillus and might therefore not be able to clear Aspergillus efficiently. Therefore, what we have done is to add Aspergillus to cells, to immune cells from CF patients, and then to see if they are able to take up the Aspergillus spores and able to kill the Aspergillus spores efficiently. And we have done that. We have been able to do that by the truly excellent facilities the University of Aberdeen has to offer, and through the funding of the Chloe Fund. What we have seen is that actually a CF immune cell is able to ingest and kill Aspergillus efficiently. Using live cell video microscopy, we have been able to look in great detail the response of immune cells against Aspergillus conidium. And you can see this here in this movie, where a very reactive immune cell from a healthy control takes up all the Aspergillus conidia or spores around him. And you can see near the end of the movie that the Aspergillus conidia that are outside of the cell start to germinate, start to grow into hyphal structures, which is the first process leading to disease. This part of my research has been published last year in the Journal of Visualized Experiments. So we have shown that immune cells from cystic fibrosis patients do ingest and kill Aspergillus spores efficiently. However, what we do see is that they mount an excessive inflammatory response, up to 4-fold higher than what we see in healthy controls. And this is what you can see here, where a CF immune cell, he takes up an Aspergillus spore, but generates an excessive inflammatory response, which we have shown also correlates to lung function within these patients and to the amount that these patients get sick. So the more responsive these cells are, the more these people tend to get sick and the lower their lung function seems to be. We have recently published these findings in European Respiratory Journal Open Research. For the last part of my research, we have been taking immune cells from cystic fibrosis patients and from healthy controls and isolate the proteins from them and then break them apart into peptides, so that using a mass spectrometer, we can exactly see how the cells from CF patients are actually different compared to those from a healthy control. And what we believe is that this will allow us to see why these cells are so hyper-responsive against Aspergillus. Our new insights obtained are highly relevant when thinking about how to prevent Aspergillus infections in cystic fibrosis and how to treat it. The WARS group will continue with this research and also look at anti-inflammatory therapies as a measure of treating Aspergillus disease in CF. So I'm hugely grateful for the Chloe Fund, which has allowed me to use sophisticated techniques in my PhD project, and also to have been able to work in the MRC Center for Medical Mycology, which is truly a great place to work.